Hello students. In this session, we are going to study about the proteins. Proteins are very important part from biomolecules. Proteins are the most abundant biomolecules of the living system and they are the fundamental structural material of animal bodies. And when the proteins are in the form of enzymes, they play a very important role in all physiological reactions. The sources of proteins are milk, cheese, pulses, peanuts, fish, meat, etc. And all these proteins they occur in every parts of the body and they form the fundamental basis of structure and functions of life. They are also required for growth and maintenance of body. And the word protein is derived from Greek word proteos, which means primary, or what we can say these are the prime importance. Proteins are polypeptide. See these peptides, these are the compound consisting of two or more amino acid, and when these amino acids linked in a chain. See these are the amino acids, and when these amino acids are linked in a chain, they form proteins. And proteins are linear chain of amino acids linked by peptide bond. The bond which present in protein is C double bond O and H. This is called as peptide bond, and each protein is a polymer of amino acids. So these are the proteins which we get from amino acids. By the removal of H2O from amino acids, these amino acids forms peptide linkage C double bond O NH. It is also called as peptidation, and this is the formation of protein. So what are the amino acids? These amino acids are colorless crystalline solids and water soluble high melting solids, and they behave like salts rather than the simple amines or carboxylic acid. Now, this is a structure for alpha amino acids. This is a carbon which is alpha carbon atom, and this is carboxylic group. C double bond O, OH group, carboxylic carbon. This is alpha carbon atom. On alpha carbon atom, presence of amino group, NH2 group. And therefore, these are referred as alpha amino acids. Carbon atom, alpha carbon atom, which is bonded with the carboxylic group, NH2 group, hydrogen, and one is the R group, that is a side chain. In case of these alpha amino acids, on that alpha carbon atom, amine group and carboxylic group are present, and hydrogen which is present on alpha carbon atom. These three groups are fixed. Okay, so what is the change? The R group is variable. That R group it change and it form the different structure for these alpha amino acids. But this NH2 group and COH group and hydrogen on alpha carbon atom, these are fixed, and this is a structure for alpha amino acids. And these proteins are formed by the linear chain of amino acids by the linkage of the peptide bond, and therefore these amino acids, these are called as monomers. Okay, this one alpha amino acids, another one alpha amino acids, these are called as monomers. Means that monomer, it is a molecule that can be bonded to other identical molecule and they form the polymer, and here they form the protein polymer. All these proteins are polymer of alpha amino acids. Amino acid contain NH2 group and carboxylic group as a functional group, and depending upon the relative position of amino group, means on which carbon atom it is present with respect to the carboxylic group, these amino acids are classified as alpha, beta, gamma. But for these proteins, we are related it here only with the alpha amino acids, on which that NH2 group is present on alpha carbon atom, and only alpha amino acids are obtained on hydrolysis of proteins. This is for the classification of amino acids. In case of these alpha amino acids, if they contain equal number of amino and carboxylic group, so it makes the neutral structure, neutral alpha amino acids. If more number of amino group than carboxylic groups, they makes the basic alpha amino acids. And if they contain the more carboxylic group as compared to the amino group, it makes acidic in nature. Means this is the neutral alpha amino acids, basic alpha amino acids, and acidic amino acids. These are the three classified group of amino acids. And the amino acids which are synthesized in the body, these are known as non-essential amino acids. And on the other hand, those which cannot be synthesized in the body and we must obtain through our diet, these are known as essential amino acids. Now see, this non-essential and essential amino acids, these are either neutral, basic or acidic. This is a not different examples for this. These are among from the neutral, basic and acidic amino acids. So see here the structure for these neutral, acidic and basic amino acids forming these proteins may be characterized as neutral, acidic and basic depending upon the character of the side chain. This is alpha carbon atom on which NH2 and COH group and hydrogen, these are fixed. Okay, but what is here? R group is CH3. As they contain one NH2 and one COH group, these are neutral. In acidic alpha amino acid, see this is the alpha carbon atom on which NH2, hydrogen, C double bond OH group are present. But here R, that is side chain, is present here. It is a CH2 C double bond OH. As they contain acidic group in the alkyl group in the R structure, these are referred as acidic amino acids. For the basics here, this is alpha carbon atom and on the alpha carbon atom, R group is CH2, 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 CH2 and NH2. They contain one more NH2 group in their structure and therefore these are under the heading of basic alpha amino acids. And these are the example for neutral, acidic and basic alpha amino acids depending upon the character of side chain which is attached on the alpha carbon atom. These are the example for neutral alpha amino acids. There are total 20 alpha amino acids. Out of these, 15 are neutral and these are the example glycine, alanine, vanillin, leucine, isoleucine, asparagine. These are the example for neutral alpha amino acids. Now see in their structure for these alpha amino acids which are neutral, they contain one COH group and one NH2 group in their structure and therefore these are referred as neutral structures for alpha amino acids. These three examples for basic alpha amino acids where R contain an amine functional group. The amino acid produces basic solution because the extra amine group is not neutralized by acid group. And see here these amino acids which have basic side chain, they include lysine, arginine and histidine. The structure for basic amino acids, see this is the alpha carbon atom on which NH2 carboxylic group and hydrogen is present and this is the R group. That R group contain NH2 structure. Here also CH2 4 time and NH2, it is R group which also contain NH2 group and therefore these are referred as basic. Here also R group contain NH and histidine, it is example for all these examples lysine, arginine, these are the example for basic amino acids. All these basic amino acids are essential amino acids, means which you have to require here to introduce these proteins to our diet. These are the example for acidic amino acids which contain one more carboxylic group in their structure. Aspartic acid and glutamic acid example for the acidic alpha amino acids where R group it contains CH2 C double bond OH. Here also it contains C double bond OH group. Means these are the example for acidic amino acids. All these alpha amino acids have their travel names which usually reflects the property of that compound or its source. And therefore all these amino acids, they are named according to their abbreviations like by using the 3 letters and by using 1 letter symbol. See, suppose here for this glycine, its short form is G. It is that is 1 letter symbol. It is also referred by the 3 letter symbol GLY. Okay, and the side chain is H. For the histidine, the short letter that is 1 letter is H. It is referred by the 3 letter symbol HIS and this is the side chain which is present on the alpha carbon atom. So all these groups are side chain which are present on this alpha carbon atom, that is R group. This is the name for amino acids. This is 1 letter symbol and this is 3-letter symbols. So this is the, for this, suppose here for the serine, it is letter is, one letter is S, 3-letter symbol SER, and the side chain, or what you can see here, R group is CH2OH. For the valine, V, VAL, and R group is CH3 twice. So this is for the, some more example for the alpha amino acids. In all these alpha amino acids, all they contain NH2COH group H and R, but only for the glycine, that R is equal to H. R group is H, and therefore except glycine, all other naturally occurring alpha amino acids are optically active. But in case of this glycine, the alpha carbon atom is asymmetric. This is the alpha carbon atom, and this is the alpha carbon atom is here, what it having, non-asymmetric carbon atom. It is no asymmetric carbon atom, and therefore it is optically inactive. Glycine, it's named as it is sweet in taste, and therefore in Greek world it is referred as a glycos. And triosine was the first obtained from the cheese, which is example for these alpha amino acids. Most naturally occurring amino acids have L configuration, and the L configuration is, that is the position of this NH2 group, which is written on this left side, and that is referred as L amino acids. So remember here glycine, which is optically inactive, only glycine alpha amino acid which is optically inactive, as it not contain asymmetric carbon atom. As on this carbon atom, both the two groups are same. Hydrogen, two groups are same, and therefore that glycine, it is optically inactive. Otherwise, all these alpha amino acids are optically active. The optical activity of these amino acids, and the next point is about the amino acids are essential and non-essentials. Non-essential amino acids, amino acid that our body make it, these are the non-essential amino acids, and the alpha amino acids which we get them through our diet or food, these are called as essential amino acids. Therefore these are also classified as essential and non-essential amino acids. These are the example for these essential and non-essential amino acids. Histidine, isoleucine, leucine. These are the example for essential, and these are the example for non-essential amino acids. So what we can see here, these proteins on hydrolysis gives mixture of alpha amino acids. As on hydrolysis it gives a mixture of alpha amino acids. If you want to prepare these proteins, what we require, amino acids, and by the removal of H2O from amino acids, we get the proteins in the form of peptide bond. So this is a structure for proteins, C double bond O NH linkage, which we get from amino acids. Amino acids, C double bond O OH, OH group from this carboxylic group, and hydrogen from NH2 group. Removal of H2O takes place. This is the one amino acid molecule. This is the another amino acid molecule. And by the removal of H2O, we get here proteins which contain C double bond O NH linkage, peptide linkage. Now see how this is about the Zwitter ion. This is a structure for alpha amino acids, R, C double bond OH, and NH2 group. NH2 group having on that nitrogen lone pair of electron. So carboxylic group hydrogen, and so this lone pair of electron take one proton in aqueous medium, and it form NH3 plus and C double bond OO minus. And that behavior of these is due to the presence of both acidic and basic group in the same molecule. And therefore this carboxylic group lose a proton, and this proton is accepted by the amine group, and therefore what they form here, the structure of the Zwitter ion. In the Zwitter ion form, amine acid shows amphoteric behavior as they reacts with the both acid and base. So how we can define here Zwitter ion? In aqueous solution, the carboxylic group can lose a proton and amine group can accept a proton. And what they gives here, dipolar ion, which is known as Zwitter ion. See, this is a Zwitter ion which contain the dipole. One is the negative pole and one is the positive pole. And this is a neutral as they contain both positive and negative charges. Alpha amino acids are water crystalline solids, means they not behave like simple amines or carboxylic group. And the properties of these alpha amino acids are due to the formation of a peculiar structure of this Zwitter ion. See, this is a carboxylic group, NH2 group, one proton of the carboxylic group, it form NH3 group, and therefore NH3 it form the positively charged pole, and C double bond O negatively charged pole. So this is about the Zwitter ion structure. In these three Zwitter ion structure, amino acid exists in the different forms depending upon the pH of the aqueous solution in which they dissolve. Means if we dissolve this amino acid, so in the water in the aqueous solution, what they shows a change in the pH. First, we see this is a structure for the alpha amino acids in which hydrogen of this carboxylic group, it forms bonding with this NH2, it form NH3 structure. So it form anion as the negative charge is on C double bond O minus. It form the anion structure. And when that proton it forms NH3, it forms Zwitter ion structure. And in presence of this Zwitter, in presence of H plus ion, Zwitter ion, it converted into C double bond OH structure. This proton is accepted by the negatively pole, the C double bond O, and it forms the cation. Now see here that on that NH3, it forms the plus charge, it is cation, and therefore it is acidic in nature. And as it is acidic in nature, it shows a very low pH. pH is less than 2. Why it is acidic? Because on NH3 it is a positively charged. Then it is a cation, and therefore overall charge is plus 1 charge on the cation. Fourth is Zwitter ion, it having two poles, C double bond O minus, NH3 plus, and therefore it behave like a salt, and therefore it is neutral in nature. pH is near to the 6, and it having no net charge on the Zwitter ion. Here for the C double, when it removes one proton from the carboxylic group, it forms C double bond O minus, means it is anion. As it having the negative charge, it is basic in nature, and therefore it shows a high pH value. pH is greater than 10. It behave like a basic in nature, and the overall charge is -1. So this is the amino acid which exists in three different form depending upon the pH of the aqueous solution in which it is dissolved. It shows the cation, Zwitter ion, and anion. One is the acidic nature, neutral nature, and basic in nature. So this is about the types of proteins. Based on the molecular shape of proteins, they are classified into two types, globular protein and fibrous proteins. First, we see about the globular proteins. Their molecules of these globular proteins are spherical in shape, and now see, as they shows the spherical in shape, the structure is result from coiling around of the polypeptide chain. As we have studied how the proteins are formed by the peptide linkage. So when they form polypeptide chain, that the coiling is takes place, and due to that they form the globular structure. And these globular proteins are soluble in water, acid, base, and salts. Whereas for the fibrous proteins, these molecules are in the form of fiber-like structure, rod-like shape, or what you can say, these are elongated. And when this polypeptide, that is a polymer, as proteins are polymer, they are made by the monomer of the amino acid. So when they form polypeptide chain, it runs parallel and held together by hydrogen or disulfide bonds. And therefore they form the fiber-like structure. And these fibrous proteins are insoluble in water, but they get soluble in strong acids and bases. So depending upon their molecular shape, that one is in the spherical shape, one is in the fiber like shape, they are classified into globular proteins and fibrous proteins. Insulin, egg albumin, or legumulin, casein of milk, these are the example for globular protein, whereas keratin, which is present in hair, nail, or myosin, which is present that proteins of muscles, which is present in the muscles, myosin. These are the example for the fibrous protein. So this is about the classification on the basis of their molecular shape. They are classified into globular protein and fibrous proteins. So this is all about the alpha amino acids, classification of alpha amino acids, and types of proteins, about the these proteins. Okay, so this is for the some study of the protein points. Thank you.